In this topic, we're going to look at plant tissues. So by the end of this topic, you'll be able to identify and label the different tissues found in plants. So what are the different plant tissues? You've got your meristem, parenchyma, collenchyma, sclerenchyma, your vascular tissue, which is your xylem and phloem, and then your epidermis. So we're going to have a look at meristem first. Meristematic tissues are localized regions of cell division. In plants, these are specific regions, namely the apical meristems, which you find in the tips of growing roots and shoots, and the lateral meristems, which you find in your cambium, and the cambium is found between the xylem and the phloem. So we know that meristematic tissue is actively dividing. So how does it divide? It divides by mitosis. So we'll discuss mitosis in a later topic. The shoot apical meristem is at the tip of every growing shoot. So you can see this at the top of the photo. It's a region of actively dividing cells, the very top. Behind those cells, you've got cells that are growing by taking in water. And behind those, you've got older cells that are starting to specialize into xylem and phloem, which is your vascular tissue, and then the epidermis. So here you can see a shoot apical meristem. Notice how the cells are unspecialized towards the top and they're actively dividing. In the root apical meristem, you've got a root cap, and behind the root cap, you've got actively dividing cells. Now, why do we have a root cap? Why does a plant have a root cap? The root cap is made of unspecialized cells that protect the root as it drives its way through the soil. The arrow on the left points towards cells that are dividing by mitosis. Notice how the nuclei have been stained red. So they've been stained because the chromosomes are actively dividing at this time. And lateral meristems, which you'll find in woody plants. So every season the xylem and phloem are remade. The old xylem becomes wood, which is why you can cut a tree in half and work out how old it is by counting the number of rings. Here you can see the arrows pointing towards about 2 layers of cells, and these make up your cambium. So the cambium you find between your xylem and phloem, and it's found in the vascular bundle. So the cambium, you'll notice where the arrow is pointing towards, is between the xylem, which is at the bottom, and the phloem just above it. And just above the phloem, you'll see those cells that have got thickened red walls. So they've been thickened with lignin. So just above that's a sclerenchyma cap. So you've got sclerenchyma, phloem, cambium, and then xylem below that. So let's look at the simple tissues, which consist of one cell type. You've got parenchyma, and these are thin-walled and they're alive at maturity. Collenchyma are thick-walled, they're also alive at maturity. And then your sclerenchyma are thick-walled, but they're dead at maturity. They've been thickened with lignin that is impermeable. Your epidermis can be modified to form trichomes and root hairs, and your epidermis is also alive when it's mature. So let's look at parenchyma. Parenchyma cells have got thin walls, and these are used as packing tissue. So a good way to remember is parenchyma equals packing, PP. There are a whole lot of regular shaped cells that are clumped together. You'll notice in these cells that there are gaps between the cell walls, which is typical of parenchyma. You'll also notice that these cells have got areas, those pink dots, that have been stained. These are probably storage areas in root containing starch. So something to remember about parenchyma, thin walls, and they've got gaps between the cells. Now, when you look at collenchyma have got thickened walls that have got extracellulose laid down, particularly in the corners. This is why you've got to get a circular effect in the middle. And this circular effect in the middle is living cytoplasm. The cytoplasm is still active because the cell walls are freely permeable. The effect is that the cells look round. Now, normally cellulose stains either blue or green, but this cellulose has been stained red. We know that it's cellulose because if you look at the epidermis at the top, it's also red around the outside. And we know that cellulose walls are found in the epidermal cells. How do we know that this is Collenchyma? This is because the cells are round and the edges are thickened with cellulose. Now, what does Collenchyma do? Collenchyma helps with support. So if we look at the different tissues in the context of a stem, A is your epidermis, B is Collenchyma, which you'll find all the way around the edge of a stem, and this is for support. Region C, large cells, thin walls with gaps between them. So those must be your parenchyma. And then, sorry, that's D. C is vascular tissue. So A is epidermis, B is Collenchyma, C is your vascular tissue, and then D is parenchyma. So moving on to Sclerenchyma. Sclerenchyma cells are modified cells with thickened cell walls, and they've been thickened with lignin. The cell contents will eventually die. You find Sclerenchyma in sclerids and fibers, which you find in vascular tissue. So you've got something called a Sclerenchyma cap, which is what the arrow is pointing towards. It's often associated with vascular bundles and dicotyledons. The Sclerenchyma cap is the top layer of cells, and it has lignin that's been stained red. So you can easily identify Sclerenchyma because of the lignin, thick lignin. At the bottom, this is xylem. How do we know it's xylem? Because there's also lignin and the cells are much larger. Now looking at the Sclerenchyma cap, the Sclerenchyma cap is for support. So imagine that the xylem and phloem are like drain pipes going next to the Sclerenchyma cap, which is made of a brick pillar. Looking at complex tissue, you have got xylem, which conducts water, and then you've got phloem, which conducts food, which is sucrose. You might also come across the periderm, which is a protective covering. Okay, vascular tissue, xylem and phloem. Here's a slide showing xylem and phloem. You'll remember from IGCSE that if you cut a stem open, the phloem is on the outside and the xylem is on the inside. How to remember this is if you ring bark a plant or a tree, so you cut the outside, the tree will eventually die. This is because it can't transport sugars in the phloem. The xylem is made of elongated cells. You've got the cambium in the middle, and then you've got your phloem on the outside. And the phloem's got sieve plates between them. Here's a nice example of xylem. It shows tracheids and xylem vessels. The xylem vessels are wider and they're grey in colour. In this example of xylem, you'll notice little holes in the xylem. These are called pits. We'll look at xylem in a little bit more detail when we get to transport in plants. Now, xylem needs to be thickened to help with support. The young xylem, which is what you can see on the left, has got spiral thickening. And as the cell gets older, so more lignin is laid down until eventually you get a cell that looks like the one on the very right. And it's got tiny little holes in it called pits. Phloem has got sieve plates between them. And it's also got companion cells sitting next to them. So looking at this leaf, you'll notice the vascular bundles. Notice the parenchyma at the top, those large cells, thin walls, air spaces between them. And then you've got the xylem. How do we know it's xylem? It's got lignin walls which have been stained red. And then just beneath the xylem, you've got the cambium, which is about 2 cells thick, and it's found between the xylem and phloem. And here you have the phloem. The phloem has got cells, well, medium-sized cells with tiny cells squashed between them. So here's a section through a root. Here you can quite clearly see the phloem. These are large cells with tiny cells squashed between them. What do you think this shows? This shows xylem. How do we know it's xylem? It's got lignin walls which have been stained red. And here you have phloem and xylem. This shows xylem and phloem in longitudinal section. You can see the xylem, which are your tracheids and xylem vessels, have different types of thickening of lignin. And then you've got your phloem. And the phloem are elongated cells placed end to end. Here's a beautiful example of tracheids, which you find in xylem. Notice the lignin walls that are quite thick, and these form long tubes through the plants. Okay, let's move on to the epidermis. All structures are covered with a thin layer of cells called epidermal cells. Notice the thin walls and how the cells are packed together with no air spaces between them. You'll also see the nuclei. Now the epidermis can be modified to form guard cells which surround a stoma, trichomes which might be glandular. These might secrete a scent or they might secrete poisons to stop animals from eating the plants. And then you've got the root hair cells. Here you can see epidermal cells with guard cells surrounding stomata. This is the underside of a leaf. Also notice how the cells are packed tightly together like a jigsaw puzzle. And here you've got a guard cell surrounding a stoma. Now in this picture you've got epidermal cells that have been modified to form hairs. These hairs are probably to protect the leaf from drying out. And in this picture you've got epidermal cells that have been modified probably for glandular, secreting some sort of scent. So what have we learned in this lesson? We've discussed the different tissues. You've got your meristem, parenchyma, sclerenchyma, collenchyma, vascular tissue and epidermal tissue. And that concludes our lesson. The end.